Hello, everyone. We're going to have a lecture on Unit 3. The topic is blood typing and blood transfusion. So for this week, we're going to have 3 lectures. The first part will be the blood typing and blood transfusion. The second part will be the lymphatic system. And the third part will be the immune system. That is for the week of March 22nd to March 29. Okay, so this lecture is the first part, is blood typing and blood transfusion, and you really need to first read your textbook from page 663 to 666. You need to read the textbook, those pages first, and then watch this video. Okay, let me share my screen with you. All right, so blood typing and blood transfusion. Okay, before we start, there are some terms you need to know. Number one is antigen. When you talk about antigen, it's something big. It can be a protein, it can be a sugar molecule. And this antigen can trigger, this substance can trigger the protective defense mechanisms. That's called antigen. On the blood cells in your blood, the antigens can determine your blood type. In that case, these antigens are proteins. They are located on the surface of red blood cells. When you talk about antibodies, antibodies are all proteins. They're produced by plasma cells. Okay, plasma cells are the derivatives from B lymphocytes. If you remember, the one white blood type, one white blood cell is called B lymphocytes. When B lymphocytes get mature and differentiate, it can become plasma cell. Okay, so here we go, the blood typing. Blood type is a classification determined by the presence or absence of specific surface antigens in red blood cell membranes. Okay, there are about 50 different antigens on the RBCs, but usually for blood type, we use the most common ones to determine the blood types. Okay, that is antigen A, antigen B. So you heard about type A blood, type B blood, and type O and type AB. So here we go, the blood types. All right, so if you notice, if it's type A blood, there's antigen A present on the RBC, the second column. All right, antigen. Now, if you look at a type B blood, then on the RBC surface, you have B antigen. All right, next one. If it's a type AB blood, that means on this RBC surface, you have both antigen A and antigen B. Next one, type O blood. Okay, if it's type O blood on the RBC, there's no antigen A, there's no antigen B. Okay, so so far, just by looking at a column 1 and 2, you can see the pattern. Whatever antigen you have on the RBC, then the blood type is related to that antigen. So if you have A antigen on the RBC, then it's type A blood. If you have B antigen, then it's type B blood. So we can say antigen, the type of antigen determines the blood type. Okay, if it's on the RBC surface, there's no antigen A, there's no antigen B, then it is called type O or type 0. All right, now look at the column 3, antibodies. All right, normally antibodies are circulating in the plasma, the blood plasma. Okay, so you have to remember this. Antigens are located on the cell surface. Antibodies are in the plasma, blood plasma. Okay. In general, the antibodies are, in general, you will not have antibodies unless you are exposed to a specific antigen. For example, if you have a flu, okay, so you are exposed to certain viruses, then your immune system will start to produce antibodies against this specific antigen. So before you are exposed to the antigen, you do not have that antibody in your blood. Okay, that's in general. But however, in blood, there's an exception. Okay, if you have type A blood, you already have anti-B antibodies circulating in your plasma. This is a given. This part has been done during the fetal development. You just have to memorize this, okay? If you have type A blood, you have anti-B antibodies. If you have a type B blood, then you have anti-A antibodies, all right? If you have type AB blood, you do not have either anti-A or anti-B antibodies. If you have type O blood, then you have both anti-A and anti-B antibodies. For the Rh one, I'm coming back later. So let's just look at the basic ones. Type AB, type A, type B, type AB, and type O blood. Okay, so we talked about the basic blood types, and we said the antigen determines the blood type. Okay, so when we are doing blood transfusion, we want to make sure the donor's RBCs will survive in the recipient's blood. Okay, when we gave blood, if you are a donor, you gave blood, what you're giving, the most important part is your red blood cells. You want the donor's red blood cells survive in the patient's blood. Okay, so we have to test for compatibilities, to make sure the donor's RBCs can survive in the recipient's blood. Okay, all right, let me use the whiteboard to explain this part, and then we'll come back to this slide. Okay. All right, let's first review the blood type and antibodies. Let me see, how do I? Okay, so here we go. I don't know, how can I get rid of this? Hold on. Here we go. I like that. Okay, so let's review this. Okay, so if I draw a RBC, okay? So this is my blood, all right, Joy's blood, okay? So if I have antigen A on the cell membrane, then my blood type is what? Type A, is it? Okay, so now there's another person's RBC here. Let's say Alex, right? And it has B antigen on the cell surface. Then this blood type is B. Very good. Then the third person, okay, RBC here. All right, let's say this is John. On this person's RBC, it has both antigen A and antigen B. So what blood type is that? AB, very good. The last one, RBC here. Okay, so we say on this person, no A, no B. So this person's blood is 0, type 0, that's type O. So antigen determines the blood type. That's the first important sentence you need to remember, okay? Antigen, type of antigen determines the blood type. All right, if you have type A blood, then during the fetal development, you already developed anti-B. Oops, anti-B. Antibody. So this is a given. If you have type A blood, you always have anti-B antibody in your blood plasma, okay? If you have type B blood, then you have anti-A antibody in your blood plasma, okay? If you have type AB blood, then you have no anti-A or anti-B antibodies. You don't have either one. Okay, no. If you have type O blood, then you have both anti-A and anti-B antibodies. So you notice on my drawing, you use this capital letter to represent antigen. Then you use the anti-A or anti-something to represent the antibody. Okay, so, all right, let me repeat this. Number one important thing is antigens are on the surface of RBC. Antigens determines the blood type. Number two is antibodies are in the blood plasma. If you have type A blood, then you have anti-B antibody. Okay. Number three, antigen only react with specific antibody. So only anti-A antibody react with A antigen. This reaction will cause a process called agglutination. That means forming the clumps. So if I draw this, that's like a RBC with A antigen react with anti-A antibody. Then what's going to happen to this cell? The cell is going to lyse and it clumps together. That will destroy this cell. You got that? Okay, so if someone has A antigen on RBC in his or her blood, you cannot have anti-A antibodies. because the antibody will destroy all of these RBCs. Okay, let me see, how do I clear this? I need to clear this page. Hold on. Right, board. Oh, it's gone. Oh, okay. Alrighty. So now those are the basics. Okay. So if we go back to my slide to review before we and type AB blood will have both antigen A and antigen B, and it will not have either anti-A or anti-B antibodies in the plasma. All right, this part you have to memorize, okay? All right, now we can continue. Okay, so here we go. Now we're talking about blood transfusion. Okay, blood transfusion. So we're going to have a donor here, which will give blood to somebody. Then we have the recipient, okay, the patient on this side. Okay, so the question is, if a patient has type A blood, if the patient has type A blood, this patient can receive what type of blood? Okay, so one more time. If the patient has type A blood, who can be the donors? What blood type donors can this patient use? Okay, so how do we solve this problem? Remember, I said, when you gave somebody blood, you want to make sure the donor's RBCs are going to be able to survive in patient's blood. Okay, how can we make sure this donor's RBC survive in the patient's blood? That means this patient cannot have antibody which react with the donor's RBC. One more time, the patient's antibody cannot react with donor's antigen. All right? If this antibody can react with this antigen, then you're going to destroy all of these RBCs. Then you did this transfusion for nothing. Okay? You want to make sure this donor's RBC will work in the patient's blood. To make sure this RBC will work on this patient's blood, you want to make sure that the patient's antibody does not match this RBC antigen. does not match the antigens on the donor's RBC. Okay, let me give you an example here. Okay, so let's say if the donor has an RBC, if the donor has type A blood, which has A antigen on the surface, and we already know the patient has type A blood. So in the patient side, in the plasma, what antibody this patient's blood has? Is that anti-A antibody? No, because anti-A will react with its own antigen, right? So it has to have anti-B antibody in the plasma. Okay, so if the donor has type A blood, then this RBC get to the patient's blood. This antibody right here cannot react with RBC with antigen A on the surface. So this will work. This will work. Okay, let's try another one. All right, if this donor RBC has a B antigen on the surface, then you put this RBC into the patient's blood. Okay, what's going to happen is this RBC has a B antigen, and then see what is that. Okay, this type anti-B matches this B antigen and react with it and destroy the cell. So the type B blood donor cannot be used for this patient. Okay, let's try one more. Donor, patient. Okay, so if the donor has A and B, that means this donor is type what blood? AB. Very good. Okay, the patient has type A blood. So the question is, the type AB blood, can this person with type AB blood give blood to this patient with type A blood? That is the question. Okay, so remember, we want to make sure this donor's RBC can survive in the patient's blood. So we know this RBC has both antigen A and antigen B. We also know if this patient has type A blood, then it's going to have type anti-B antibodies in his plasma. All right, so if you have anti-B antibodies and specific antibody react with a specific antigen, so is this going to have a reaction here? Yes, and that will destroy all these donated RBCs. Then it's not going to work. Okay, one more. Oops, let me change color. Donor's RBC is type O blood. What does that mean? No A, no B antigens on the surface. Can this blood be used on this patient? Okay, let's look. This RBC doesn't have A, doesn't have B. On the patient side, it has anti-B antibody. So this antibody will try to find B antigen and react to it. But since there's no B antigen on the surface of RBC, so there's no reaction. So this will work. Okay? All right. So let's try one more time. Okay, this is donor's RBC. This is the donor side. This is the patient side. Okay, the question is, if the patient has type A blood, who can be his or her donors? Okay, so how can we do that? First, we can actually write out all the possibilities, all blood type possibilities. That's type A, type B, type AB, and type O. And then we know the antigen determines the type. So if we write this capital A, letter A, that means this donor has type A blood. Okay? And on the patient side, all right, if this patient has type A blood, that means this patient has anti-B antibodies in the plasma. Okay? So out of these 4 possibilities, which one can be used for this patient? Okay, as long as there's no reaction between this anti-B on this donor side, then it's good. But these 2 I'm drawing here, this anti-B antibody definitely can react with type AB blood, because here on the surface you do have the B antigen. All right, so this type B anti-B antibody can react to this antigen. Okay, so if this donor has B antigen type B blood, then this anti-B can react to this and destroy the cell. All right, so if the patient has type A blood, only people with type A blood or type O blood can be used as donors' blood. You got it? Okay, let's try another one. So donor on this side, patient on this side. Okay, now we say this patient has type B blood. Okay, what blood types can be used for this patient? Again, we're going to write down all the possibilities. Okay, all the possibilities. That's only these 4. All right, and then you need to remember right away, on the patient side, if it's a B blood, then you have anti-what? Antibody in this blood? Anti-A. Okay, this is given. You have to remember. All right, and we know specific antibody can react to the specific antigen and cause agglutination. Okay, that's what you want to avoid. Okay, so you want to have donors RBC. Oops, wrong. You want to have donors RBC here, which can survive in the patient's blood. Okay, all right. So now this anti-A antibody only reacts to A antigen. So you look on this donor side, which type of blood will have A antigen? Is this one and this one, right? So the type A and the type AB cannot be used in this case, because this anti-A antibody is going to react to the antigen A and destroy all the transfused blood cells. All right, let's try one more. Donor on this side, patient on this side. Okay, so now we say this patient has type O blood. Okay, so who can be his donors? Again, we're going to write all the possibilities out. All right, and then we'll think about what antibody on this side if it's type O blood. Okay, if it's type O blood, there's no antigen A, no antigen B, but you will have both anti-A and anti-B antibodies. So as long as you have A or B antigens on this side, this type of blood cannot be used because the anti-A or anti-B antibodies will react to these antigens. So only type O can be used. All right, are we good? Okay, try one more. If the patient has type AB blood, okay, who can be the donors? So again, we write down all the possibilities. All right, so if it's type AB blood, what antibodies can be found in the type AB blood? Nothing. No anti-A, no anti-B, right? All right, so if there's no anti-A or anti-B against these antigens, then all of these can be used because all of these RBCs coming to the patient's side, they can survive. There's no antibody against either one of those. Are we good? Okay, so the key to remember here is the donor's RBC cannot react with recipient's antibody. Oops, here. Yeah, where did I go? Sorry. Share. Okay, so let's come back to these slides. All right, so here, all right, donor and recipient relationship. So now let's read it. Donor cannot have antigens which bind to the antibody in the recipient's blood. That's what I was explaining on the whiteboard, okay? So donor's antigens, think about these antigens, they're on the surface of RBC, all right? They cannot react with the antibodies in the recipient's plasma. If there's a reaction, then you're going to destroy all the donor's RBCs. And then you did this transfusion for nothing. No donor's RBC can survive. Okay, second part. Recipient can receive any blood which does not have antigen to bind with its antibody. Okay, so it's the same thing. It's pretty much the other way to say that, okay? From the recipient's side, the antibody in the recipient's blood cannot react with donor's antigen. Okay, so if the antigen and antibody react together or bind together, it can cause all of the following. So like say agglutination, that's clumps together, or which can clotting the small blood vessels and block the blood flow, or it can lyse the RBCs, Okay, so destroy all the RBCs, all right, and other stuff. So basically, when you do blood transfusion, you want to make sure that there will be no antigen antibody reactions. Okay, so on your lab handout, you have a chart. You want to do a practice at home and to make sure you understand how does this work. Okay, so maybe we can try one more. Let me see, stop sharing here. Let's try to solve one more problem here. I'm using the whiteboard here now. Okay, so let's say if I say, okay, the patient is on this side, the donor is on this side. Okay, so if the patient has type A blood, who can be his donors? What type of blood can be used as donor's blood? Okay, so can you do this, everyone? The first step, write out all the possibilities. Then, what? Patient, the antibody, which has anti-B antibody in this type of blood, cannot react with antigens on the donor side. So if you have anti-B here, it's going to react with this B antigen. If you have anti-B here, you can act with this B antigen. So these two types cannot be used as donor's blood. Okay, so please practice this. There's a chart on your lab handout. Okay, do that. Okay, and let's try another one. Ask the question in a different way. Okay, if the donor is on this side, patient on this side. Okay, now the question changed a little bit. If the donor has type A blood, this blood can be used for what blood type patients? One more time. If the donor has type A blood, this type A blood can be used for patients with what type of blood? Okay, the question is asked a little bit different, but the principle is the same. The principle is you want to make sure this RBC with antigen on it can survive in this patient's blood. Hey, you stop. Sorry, my little dog. Oh, I'm so sorry. Okay. So on this patient, you want to make sure there's no anti-A antibody. If you have anti-A antibody, you're going to have reactions. Okay, so what type of blood does not have anti-A antibody? Stop. Either. Hold on 1 second. Oh. Okay, so the donor has A antigen. You want to make sure the patient does not have anti-A antibody, which can destroy all these RBCs from donor side. Okay, so the question is, which type of blood has anti-A antibody in the plasma? Again, you first write out all the possibilities. Only these 4 possibilities, right? And then you see Type A blood has what antibody? B. Type B blood has anti-A. Type AB has nothing. Type O has both. All right, so now you see this one can react with this antigen. This one can react with this antigen. So this patient cannot receive this blood. This patient cannot receive this blood. You got it? Let's try one more time. Donor here, patient here. The donor has type AB blood. Who can receive this blood? So again, you're going to write down all the possibilities and think about the main thing you want to consider is this RBC, which has both A and B antigens, can survive on the patient side. That means on the patient side, you cannot have anti-A nor the anti-B antibodies. If you have either one of them, it's going to destroy the donor's RBC. So out of these 4 types of blood, which one has no anti-A, has no anti-B either? Only this one, right? So only the patient with a type AB blood can receive blood from this source. Okay, all right. So please practice yourself, okay? Because now you probably think, okay, I got it. But if you don't practice, you're going to forget tomorrow. You get yourself confused. All right, so make sure you practice. Okay. All right, so let's look at our slides. Continue. Okay, so we were, okay, let me go back. Okay, so we talked about transfusion reactions, okay? And then you're going to try to work on this, practice on this, on the chart in your lab handout. And the next part is blood type is determined by genetics. Okay, so when we say genetics, that means it's determined by the specific genes. If you look at the genes, we have dominant genes and recessive ones. Let me explain this. Okay, so in each trait, okay, if you have both dominant genes, then it's going to express the phenotype. But if you have 2 recessive genes, only when you have 2 recessive genes together, recessive trait, if you only have a trait with both recessive genes on it, then you're going to have the recessive phenotype. Okay, let me give you an example for that. How do I do that? Okay, let me go back to the whiteboard. Okay, for example, all right, this is a trait, okay? It's a trait. Okay, so on this side of chromosome, there's a gene with a dominant A on it. On this one, you have a dominant A. That means the blood type for this person is type A blood because the A gene is dominant gene. Okay, so on this trait, if you have a dominant A, but on this one you don't have an A, this little I is recessive. Doesn't matter, because if you have, as long as you have one dominant A, it's going to give you the A type. But if you have a trait with both recessive genes on it, too small, there's no A at all, okay? In this case, you're going to have type 0, type O. Okay, try one more. Okay, this trait, you have a B, you have a recessive. Then what blood type is for this person? B. Okay. If this person has both A and B, because both A and B are dominant genes, then they will express at the same time. So this person has type AB blood. You got it? Okay. So now genetics, all right? So let's say this is the father side. Okay, you have type A and type A blood. This is type A blood, is it? Okay. On the mother side, you have type B blood, but you have to carry a recessive gene. So in this one, this father will produce sperms, okay? The types of blood sperms can be either have A or have this I. That's the sperm, okay? And for the mother, the eggs, you can have either the B or the recessive. And then when you make offsprings, this combination is random, all right? Let me redo this. Let me get a colored. Okay, do better job. Okay, so type A blood, that's the father. Okay, and then type B. Oh, I don't have a color on this one, do I? Here we go. On the mother side, you have type B blood, but the genotype is like this. You do carry a recessive gene. Okay, so when this one make a sperms, there are two types of sperms, one with A, one with I. When you make the eggs, there are two types. One is the B, one is the I. Okay, now when you make the offspring, okay, the sperm and the egg, they're going to the sperm will fertilize the egg to make a zygote. But this process is random. So this one has a chance to meet with this one has a chance to meet this one has a chance to meet this one has a chance to meet this one. So the possibility is the same, right? And then you can see the combinations for the genetic combinations for the offspring has the following. You can have this A and the B on this side. That's this one, A and this egg. All right, that's one. If this one, the sperm fertilize this egg, then you're going to have genetic makeup is like this one and this one. If this sperm fertilize this egg, then you're going to have this one, if this sperm fertilizes this egg, you can have this and this. Okay, so let me use the red for the female side. Okay, so this is, the red is the female side. Okay, so that is the 4 possibilities for the children. Okay, so if you look at this genetic makeup, if you have both A and B antigens presented here, then this is blood type AB. If you only have B here and one recessive, then this is type B blood. This one is a type A blood. This one is type O blood. So for parents, one with the type A blood, one with the type B blood, right here, type A blood, type B blood. And the offsprings can be type AB, type B, type A, and type O. Okay, think about it. If you have questions, write them down for me. Okay, so now let's go back to this slide. All right, so, okay, we talk about dominant and recessive traits. So genetics, the genes determines the blood type. Okay, oops, wrongly. All right, so now I'm going to introduce another antigen, which is called Rh antigen. Rh antigen. This is just like the type A, just like the A antigen or B antigen. It's just another one on the surface of RBC. Okay, so let me do the drawing here. Clear this part. Okay, so if there's RBC here, all right, we say if there's an A antigen, it's a type A blood. If there's a B antigen, there's a type B blood. There are about 50 different antigens on this RBC, okay? But these A and the B are most popular one. The third popular one is this Rh, okay? So this Rh is another antigen. Another antigen determines the blood type. So for this RBC, okay, because it has A antigen, B antigen, and Rh antigen, so the blood type for this person is type ABRH. All right, this Rh antigen can be presented as a D as well. All right, so you can say this person's blood type can be called ABD. We got that? Okay, because it's just too much writing on this side. Okay, we want to make it simpler by using the positive and the negative signs. Let's say if this person has Rh antigen on the surface, we just use a positive sign for it. All right, that means on this blood type for this person, we can write as AB positive. This positive represents, this sign represents this person does have Rh antigen on the cell membrane, on the RBC cell membrane. Okay, so let's try another one. So if there's RBC here, has A and has Rh. So this one, the blood type is A positive. We use this positive sign to represent the Rh antigen. If this person has A antigen on the surface, but does not have Rh, then we call it type A negative. That means only have antigen A, but does not have antigen Rh. All right? Okay. If you remember at the beginning, I said antibodies, all right, antibody are produced only when exposed to the antigen. Okay, except the anti-A, anti-B antibodies. All right, most of the antibodies, we do not have them until we are exposed to that antigen. Like the flu viruses, we don't have the antibodies. But once we're exposed to the viruses, then we're going to develop the antibodies against it. All right, so I did say the anti-A, anti-B antibodies are exceptions. They are developed during the fetal development already. But in terms of anti-Rh antibody, okay, it follows the general rule for all other antibodies. That means if a person does not have Rh antigen. Okay, so let's say this blood type is type A negative. That means this person does not have Rh antigen on the blood cell surface. If in this case, this person has no anti-Rh antibody until this person is exposed to the Rh antigen. Okay, so one more time. All right, so this person with A and Rh, that's called type what blood? A positive. All right, so now in this type blood, is this person have anti-Rh antibodies? No, it will never develop anti-Rh antibodies, because if you do have anti-Rh antibody, you're going to destroy your own cells. Okay, now next one, if this person has type A negative blood, can this person develop anti-Rh antibody? Yes. When this person is exposed to this Rh antigen, then it can develop anti-Rh antibody. All right, it's possible. It will. All right, so it doesn't matter if you have anti-Rh antibody in the plasma, because you don't have this Rh antigen on the surface, and it's not going to affect your RBCs. Okay, let's try one more. If this person has both A and B antigens on the RBC, the blood type for this person is AB and then Rh here, positive. Okay, so can this person ever develop anti-Rh antibody? big no-no, all right? Because if you do have anti-Rh antibody, you're going to destroy your own cells, okay? If this person has type AB negative blood, can this person develop anti-Rh antibody? Yes. Only when this person's blood exposed to Rh antigen, then is going to trigger the production of anti-Rh antibodies. Okay, so now we can come back to the slide. Okay, so now we're talking about the Rh mother carries, Rh negative mother carries Rh positive baby. Okay, is that possible? Yes. Maybe let me try this on my whiteboard here. Okay, so here, the mother, the trait is Rh positive. Oh, hold on. Yeah, wrong. Well, we can try this. Rh, that's the trait, okay? The positive is definitely the, let me redo this. Okay, the mother, the father, okay? If the mother is Rh positive, but remember the positive is dominant, negative is recessive, okay? So genotype for the mother is like that. For father, it's like that as well. Okay, so is that possible to have a child with Rh negative blood? Yes. Yes, because if the egg with this genotype, the sperm with this genotype, they combine together to make a zygote, you're going to have the genotype like this. That's two recessive genes together. So this is Rh negative blood. You got it? Okay. So let's try another one. If the mother has Rh negative blood, the father has Rh positive blood, like that. And then the child, can the child has Rh positive blood? Yes, because this negative and this positive together, you're going to have a positive Rh baby. Okay, so now we know for this Rh negative blood, can this person has ability to develop anti-Rh antibody? Yes, when this person is exposed to Rh antigen, it can develop anti-Rh antibody. It does have the ability to do that. If that's the case, then this antibody can go through the placenta and get to the fetus side and destroy the RBCs, because on this baby, on the surface, you do have Rh antigen. If you have antibody against it, it's going to destroy the fetal RBCs. This is fatal, okay? So you do not want to have this happen. This cannot happen. Okay, so one more time. On the mother side, the father side, if the mother has Rh negative blood, the father has Rh positive blood, then you can make a Rh positive baby. All right. So because mother is Rh negative, the mother has the ability to develop anti-Rh antibody. If the mother developed the anti-Rh antibody, this antibody is small enough to go through placenta and get to the fetus side and destroy the fetal RBCs. So you don't want to have this happen, right? What can you do? Okay, all right. So first, we know as long as the mother does not expose to the Rh antigen, then it will not develop the antibody. All right. So usually the first pregnancy is safe, right? There's no chance for the fetal blood and maternal blood to cross from each other. Okay, because you do have a placenta, not direct in contact both sides. All right, but at the delivering time, okay, there's a rupture, blood vessel ruptures. Then there's a chance for the maternal blood to be exposed to the fetal blood. Then the maternal side will start to have anti-Rh antibodies developed, right? So if it's for the second pregnancy, then, if the fetus is Rh positive, then there's a big chance that the maternal blood can destroy the fetal RBCs. All right. So to avoid this to happen, we can give artificial antibodies to the mother, artificial anti-Rh antibodies, a huge dose or doses of anti-Rh antibodies to the maternal blood. These antibodies are man-made, they're really big, they cannot cross the placenta, okay? At the same time, the huge doses of these artificial anti-Rh antibodies will inhibit the mother to produce anti-Rh antibodies by her side, all right, because your immune system is sensed that you have enough of that antibodies produced, then you don't produce anymore. So this way is going to block the anti-Rh antibodies going on the fetal side. All right, that is the what called the Rogan shots. Okay, that is just the high dose of anti-Rh antibodies. Okay, let's see. Okay, so here, the condition, if the Rh negative mother carries Rh positive baby and the mother develop the anti-Rh antibodies and destroys the fetal RBCs, this condition is called erythroblastosis fetalis. Okay, to prevent that, the mother should give this artificial antibodies, okay, and to prevent these artificial antibodies is called Rogan shots, all right, high doses of anti-Rh antibodies. These antibodies, they cannot cross placenta, and at the same time, it's going to inhibit the mother antibody productions. Then in this way, the baby will be safe, okay? Normally, 20 years ago, 30 years ago, if the woman with Rh negative blood, and the doctor will first ask, is this your first pregnancy or the second, right? If it's the first pregnancy, then they leave the mother alone, does not do anything. But now, the practice is, as long as the mother has Rh negative blood, the Rogan shot will given, because sometimes if the mother had a miscarriage, which sometimes we don't even know. Okay, if there's a miscarriage, you now implanted embryos, it do have the fetal blood vessels and blood developed. If there's a miscarriage there and you don't even know, then there's a chance for the maternal blood to be exposed to the fetal blood. Then the maternal side can start to make antibodies already, even you think it is your first pregnancy. So at the rule now is, if the mother has the Rh negative blood, then she will be given the Rogan shots through the pregnancy. It doesn't matter if it's the first one or the second one. Okay? Alrighty. So now, if we look at the blood typing exercise, now it's more complicated. Okay, so let's try another one. The donor on this side, the patient on this side first. Patient and the donor on this side. Okay, so now we can say, if the patient have type A positive blood, then this patient can receive what type of blood? Okay, so now how do you solve that? It's more complicated than before. So if you write all the possibilities out, like what we did before, and then you try to eliminate the ones which will not work. So you're going to have both positive and negative ones, right? So how do you solve this problem? Okay, you make a 2 steps process. The first step, you just think about ABO typing. Forget about the positive and negative. Just think about A, type A blood. All right, if the patient has type A blood, what type of blood can this patient use? So we just say there are 4 types, possibilities on this side. And this one has, patient has anti-B antibodies, which will react with this one, so this cannot be used, which will react with this one, so this one cannot be used. So we know for type A blood, you only have the only choices you can use are the type A blood and type O blood. Okay, so that's the first step. The second step is you only consider the Rh factors. Okay, so on the second step, we already know, all right, so the B and AB cannot work. So it doesn't matter is it positive or negative, this B ones cannot work. The AB one cannot work. Okay, so the only four left will be the A positive, negative, the O positive, and negative. All right, so now you don't think about A, B, O anymore. You only focus on this positive. Okay, so if the patient has positive blood, is this patient ever going to be able to produce anti-RH antibodies? If you have positive blood, you will never produce anti-RH antibody because that can destroy your own RBC. If you don't have anti-RH antibody to against RH antigen, then both negative and positive blood can be used for this patient. Let's try again. Patient on this side, donor on this side. Okay, the question is, if the patient has type B blood, what types of blood can be used for him? Okay, let's say type B negative blood, right? So the first step is only talking or thinking about the ABO typing. Oh, this is B. Okay, type B blood. Forget about the negative sign for the first step. If it's type B blood, okay, what blood can be used for this person? If it's type B blood, it has anti-A antibody. Anti-A will against this one, so this one cannot be used. Anti-A will against this one, so this one cannot be used. So only B and O can be the choices, right? So now the second step, you just have the B positive, B negative, O positive, O negative left. The second step, only four choices. Now you only consider this RH. Don't consider ABO anymore. If this patient has no RH antigen, can this patient make anti-RH antibody? Yes, if this patient is exposed to the RH antigen, then it can make anti-RH antibodies. Okay, so in that case, we do not want to take a chance. We just say, if it's a negative blood, then this person may have anti-RH antibody. If you do have anti-RH antibody, then it's going to react with RH antigens. Okay, so this one cannot be used, this one cannot be used. Only the negative blood can be used as donors. You got it? All right, so you're going to practice on this, and you do have questions on the lab handout, the chart. Make sure you practice, okay? And if you have questions, bring the questions to me, and we can talk about this in our office hour part. Okay. Okay, so yeah, this one you can, yeah, we talk about that. Okay, the last part, blood typing. You know, if you do a experiment, how can you decide what blood type you have? What type of blood do you have? Okay, this is very simple, but you have to understand this. Okay, so on the two wells, okay, there are two wells. The first one, you put anti-A antibody serum, okay, kind of like a plasma. You take all the proteins out, then it's called a serum, Okay, so this one is given. In this serum, you have anti-A antibody. In this serum, you have anti-B antibody. Okay, now you put someone's blood. Let's say you put a Joy's blood, few drops here, few drops here. If you see these clumps form, that means in my blood I do have A antigen, because only A antigen can react with this anti-A antibody. Then you have the clumps. So my blood, I have A antigen. And the same thing here, clumps. That means I have B antigens as well. So what is my blood type? Type AB. I have both A and B, right? Now here, again, in the first vial, you're going to put an anti-A antibody serum in there. In the second vial, you're going to put anti-B antibody in here, okay? Now, Alex's blood, okay? So a few drops of Alex's blood in here. There's no clumps formed. That means in Alex's blood, there's no A antigen. And in this blood, there are clumps. That means he does have B antigen. So his blood is type B. Okay, so let's work on this one. Okay, so the first well is given. You put anti-A antibody serum in here, anti-B antibody serum here, and John's blood in both wells. And then you see clumps only in the first one, not the second one. So what blood type does John have? Gotta be A, type A. Does that make sense, everyone? Okay, all right. And you do have this chart in your book. And if you need to review, go back to page 665 and look at this diagram and read that part again. Okay, so I'm going to stop here. All right, this topic is about the blood typing and blood transfusion. And please review by reread your book and also by working on the chart on your lab handout. And then write down all your questions, and then we can talk about this again during the office hour. Alrighty. And then I'm going to stop this lecture now.